All right, welcome everybody to Paleo Talks and 2021. We're Happy New Year, and we're excited to be back and have Eric Scott with us today. How you doing, Eric?
Doing all right. How are you guys?
We're doing all right. We're doing all right. We're really excited to be back and hear about some of your work in Africa today, which is a little bit outside of what we've covered so far, which is really exciting.
It's a little bit outside of my whole background. When I was in graduate school, I wanted to go to East Africa and find hominins, and then got sidetracked by things like the La Brea tar pits and the ice ages and so forth. So in a way, it's coming home, but it's also very, very new and different for me.
That's fantastic. Well, before we get on to introducing you a little bit more, Eric, we'll go over to David Moscato, one of our co-hosts here, and he's going to tell us a little bit more about how the show works.
Absolutely. So welcome back, everybody, for 2021. As Blaine said, Happy New Year. It will not surprise you to hear that our format has stayed pretty much the same. We're going to run for about an hour. We're going to start with our guests giving their presentation, and then about halfway through, we're going to open it up to questions from the audience, and the second half or so will be audience question led. So when that time comes, we'll remind you to go ahead and put your questions in the comments of the Facebook video. And as always, if for any reason you can't leave a comment on Facebook, I'll be keeping an eye on the Gray Fossil Site Twitter and Instagram accounts, so you can leave a comment there if you have to. And I'm really looking forward to this. Is an exciting topic to kick off 2021 with.
Yeah, it sure is. Thank you, David. And we also have Chris Widga with us again today. And Chris will hopefully chime in as well with some questions as we go along.
Happy New Year.
Yes. Eric, I was just earlier trying to think about how long we've known each other, and we know it's been a long time. We're thinking 94 or 95. We met when I was a master's student at an SVP meeting.
Yeah.
But we work on similar things, and it's always been so fun to follow your work. And you're over there in California. And if you can just go ahead and share your first slide. And then as you share your presentation and your first slide, if you'll start telling us a little bit more about your background and how you got into paleontology in the first place.
Okay, I can do that. Because part of the reason, I'm one of those little kids who like dinosaurs. As a young boy, like 5, 6 years old, I got the How and Why Wonder Book of Dinosaurs. And so as a little kid, I wanted to be a paleontologist. But then as I grew up, you start going to school, you start getting into things like cars and meeting girls and getting a job and getting kind of sidetracked. I was a volunteer at the La Brea tar pits, but there was a period where I thought, well, this is what I'll do. I'll have a daytime job and then I'll volunteer at places like the tar pits. But then in 1981, a book came out by a scientist we're going to talk about in a few moments named Donald Johanson, which was about the skeleton Lucy. And that convinced me to start looking at hominin fossils and so forth and take my college career more seriously. And I stayed with the La Brea tar pits. They eventually hired me, and I stayed with Southern California paleontology and Western North American paleontology. So the opportunity to participate in field work in East Africa at the Hadar site was the chance of a lifetime. So being able to present this information to you is extremely special for me, because it's been a very long arc, but a very happy arc. So with that, let's go ahead and get started on this. We're going to be talking about horses and some hominids, a little bit on hominids in Eastern Africa. And so this is actually what it looks like at what's known as the first family site in Hadar, and with bones just sitting there at the surface. This is actually something that knocked my socks off when I first started going there. The ground is just covered with fossil bones. It really is a paleontological wonderland. And part of the reason for that is because of where it's located. The Afar region is in Ethiopia, And it's right here in what's known as the East Africa Rift Zone. And what you have happening here is the African plate is pulling away from the Somali subplate. So you have these two plates separating and opening up between them. And as a consequence, you have a lot of tectonic activity, and you also have a lot of volcanic activity associated with that. And so what you're looking at is the African plate and the Somali plate separating and opening up the rift zone. You also have the Arabian plate that's separating and pulling up towards the northeast, and that's what's opening up the Red Sea and the Gulf of Aden. So all of this is going on tectonically in the region, and the Afar region, the Afr Triangle, is right there smack in the middle of this because of all of this activity. So as a consequence, you have all of these amazing sedimentary exposures just wide open there. And if you look near the bottom of this one, you see what might look like off-road tracks, and they kind of are, except they aren't made by off-road vehicles. They're made by goats and occasionally camels, because this isn't the Southern California desert, this is the African desert. And all of the exposures you're looking at have the potential to have fossils in them. So if you're a paleontologist, you're drooling, presumably, at this point. What's also nice is that because this area is so tectonically active, and you had volcanic activity, you have a series of volcanic ashes that have been laid down over millions of years. And so those ashes enable scientists to actually provide reliable radiometric dates for the fossils that they're finding. There's also wildlife in the region. Some of it looks kind of like North American. Some of it looks very different from anything you might find in North America. You also have the Afar people there. The Afar people raise goats. They are nomadic pastoralists. They raise goats, and they frequently come by, and you can see them in the region most of the time. Occasionally, they even come by your tent in the morning when you're putting on your underarm deodorant, and they giggle at you, which is a little disconcerting, but also kind of sweet. But the reason that this region is so important to scientists, to geologists and paleontologists and paleoanthropologists, is because of the fossils that are contained in these sediments. In particular, one fossil that was found, a hominin fossil that was found in 1974, that probably everybody listening has heard of. This is the fossil known as Lucy, which is a 20% complete skeleton. It is still, even today, one of the most complete skeletons of an early hominin ever discovered. And it revolutionized our understanding of hominin evolution. There were still discussions about hominin evolution being driven by brain size, being driven by cranial capacity. And with the discovery of Lucy, who was clearly bipedal, but also did not have a very large brain, it forced a shift in how we interpreted what led humans to be humans and how we came to evolve into what we are today. Based on Lucy and a number of hundreds of other fossils from the same region, from the Hadar region, in 1978, Donald Johanson, the individual who discovered Lucy, and his team named a new genus of hominin, Australopithecus afarensis, characterized by having small body size, small brains, relatively primitive cranial features, but fully upright bipedality. And so this was, again, a shift in what people thought about how humans evolved. When Lucy and her ilk were proposed as a new species, if you look at what Johanson published, and this is from that book I mentioned, the book Lucy, back in 1981, this diagram shows, and the authors posited, that Australopithecus afarensis was in fact the pivot point between the lineage leading to us, the lineage leading to Homo, and all other hominins. So you had what here are called the Australopithecines, the Australopithecines, the robust Australopithecines, which some would call paranthropines. And there was this gap, though, between afarensis and the lineage leading to the perceived to lead to us. And so if you were to look at that same family tree today, you'll see that since the 1970s, 1980s, there have been any number of additional hominin species and even some genera added to the tree. But you still have this delineation where above this line at about 2.3, 2.4 million years ago, that's where you have Homo, and you have Homo from that point forward. But Australopithecus afarensis doesn't make it to that point. It goes out around 3.2, 3.0 million years, at least according to our current understanding. And so the question remained, how do you get from something like Lucy to something like our genus, Homo? How do you make that leap? Where are the fossils that bridge that gap? And with that in mind, we talk about the region that I'd like to discuss today. This is the region known as the Lady Guerraru site in Ethiopia, which is part of the Hadar complex. And you can see it mapped here in the middle of the Afar Depression. And if you look at this map, you can see Hadar down there in the lower left, and you can see the Lady Guerraru site is starred here. And if you drill in on that map, you can see again that the Lady Guerraru site is part of the Hadar complex. And this was actually back when scientists were first looking in the Hadar region and this part of East Africa. They saw these sediments, but the folding and the faulting was so complex that they saw it and they were going, I don't know. And then you start finding hominins down in Hadar proper, and all of a sudden everybody says that's where the focus should be. The faulting, if you look at the direction of the faulting, these red lines you see here over there on the right, primarily that is faulting not caused by the separation of the East African and Somali plate, but by the pulling to the northeast of the Arabian plate. And so that's what's opening up the sediments in this region. And because of that pulling, because of that opening, you do have a stratigraphic section, but you don't have stacking of the sediments that you would have in a traditional stratigraphic section. What you have is stratigraphy that moves horizontally rather than vertically. And so within this region, you have different sedimentary packages, and the volcanic ashes in the region have been used to date these sedimentary package. So you have the Bulanan package at 2.84 million years, the Gurumaha at 2.8 million years, the Leodoita around 2.67 million years, and then the Garsalu, which is younger than around 2.58 million years. And so again, you have stratigraphy, you have dating, you have context, but it's horizontal rather than the more traditional vertical. I bring that up because of this hill that you're looking at. This is a hill known as Hominin Hill or Hom Hill, and you can probably already guess why it's named that. And it's named that because of this individual, Chalachu Sium, who found a hominin fossil back in 2013, I believe. And so this was an amazing discovery. Chalachu is to be credited for his amazing persistence and his great eyesight. It's just frustrating that he doesn't see how badly the Yankees suck as a baseball team. But let's leave that aside and just go on to the actual fossil he found. This is a hominin lower jaw that he found at this site. Here's Hominin Hill again. And here, that tuft that you see is the Gurumaha tuft, which, as I mentioned, has been dated to 2.82 million years. And that tuft can be traced all along the bottom of this hill. And the hominin jaw was found in this region at the top of the hill. So that enables, again, a sedimentary constraint and a absolute dating constraint on the age of this fossil. That's why it's been placed at 2.8 million years.
Eric, I have a comment and a question.
Sure.
So it was Lucy that, and actually that exact same book that got me the most excited, I think. And it was the first book that I had in paleontology. And, you know, these hominins, and one of the things that I always sort of wondered is, where are they stored? You know, what is it like where these fossils are taken to after they're excavated out of the ground? And have you had the chance to see Lucy and some of these other fossils?
You're going to have to wait for later in the talk. But to give you, it's at the National Museums in Addis Ababa in Ethiopia. All of these materials, whether it's hominins or any of the other fossils, they go to that institution. So if you want to study the fossils, you have to go to that museum to take a look at it. You have to be approved and so forth. So all of the material stays in country, which I think is as it should be. But yes, I'm actually, right now I'm focusing on the field because field work is so cool, but I've also done some museum work, and I'll be talking about that in a few minutes. I can't speak for other places like Kenya or Tanzania, but I suspect the same thing is true there as well, that the fossils are staying in those countries and going to their major repositories.
Thank you.
In fact, this picture here was taken at the National Museum in Ethiopia, because what you see on the left is one of the classic jaws of Australopithecus afarensis. Granted, this is, I believe, a large male rather than a smaller female like Lucy, but still, this is a sizable jaw. And on the right, you see Chalachu's discovery at Hominin Hill in Lady Guerraru. And the differences should jump out at you, where you can see the deeply convex and almost swollen lower border of the Australopithecine jaw versus the straight lower border of the new discovery. You can see, hopefully, a little bit that there's a bit of a depression in the jaw over on the Australopithecine. There's a little bit of a flattening and an inset there, whereas the same part of the new jaw is actually more convex. The foramen there opens straight out at you in Australopithecines, whereas it opens towards the back of the jaw in the new discovery. And the ascending ramus of the jaw, the part that connects to the skull, it takes off behind the second molar in Australopithecines, whereas in the new discovery, it takes off behind, well behind the third molar. All of the characteristics of the new jaw that distinguish it from Australopithecus afarensis are characteristics that it shares with later members of the genus Homo. And so the argument has been that this is the earliest record of our genus, the genus Homo, at 2.8 million years. And so if you go back to this chart and you see that delineation, this fossil files right in there between the end of Australopithecus afarensis, the end of that particular lineage, and the onset of the lineage of Homo. And so whether that means they graded smoothly into one another or not is a question that still will need more fossils to resolve it. But it does suggest that what Johansen and his team proposed way back in the 70s and 80s may essentially be correct, that based on these fossils, afarensis was the pivot point. And after that, you had other Australopithecines and paranthropines branching off in one direction, and our lineage, Homo, branching off towards eventually leading through a variety of twists and turns to us. And so you have this region, which has fossils that look like this, that may not be all that spectacular, but it did produce the key fossil that now has enabled us to extend the age of our own genus back to 2.8 million years. That being said, I am not, even though hominins are cool and they're kind of what got me involved in fossils seriously, they're not the point of study for me. Because there's a whole host of other fossils that are being found in the Lady Guerraru region. And for the opportunity to discover them, I am very grateful and fortunate to know this person. This is Kay Reed from the Institute of Human Origins, and she is the president's professor, and she's also the director of the School of Human Evolution and Social Change at Arizona State University. And so she is the lead, she's the principal investigator for this entire project. And even though she's a biological anthropologist, one of her main fields of focus has been on the paleobiology of these hominins, which means that in addition to being a biological anthropologist, she's also a vertebrate paleontologist and is very interested in all of the associated animals that are found with these hominins. She is the head PI, but there are any number of other people. This is truly an international team of people, many of whom are depicted here. And this doesn't even cover some of the many, many grad students from ASU and elsewhere who are also participating in field work. So this is very, very much a team effort. And there is a huge amount of work that's been done and still a huge amount of work that needs to be done. Because these exposures are just so spectacular, they're so rich, and they've got fossils just sitting there at the surface. I can't emphasize that enough. You walk out there and, oh, look, there's fossils. And you take a couple steps and, oh, look, here's another fossil. And it's just everywhere. It is an amazingly rich. This is a bovid horn core. What's left of it. Here's another one from 2 years later. This stuff almost literally carpets the ground. This is a pig jaw, as it was discovered. I should mention that horses are a fairly major component of this. Bovids make up, this is East Africa, so bovids make up more than half of the large mammal assemblage from this region. Pigs make up another sizable proportion. Hippos make up a huge proportion. And you occasionally even do find carnivorans like this saber-toothed cat lower jaw, all from this region in Lady Guerraru, which is again part of the Hadar complex. In terms of our field days, basically we go out to the field and we just cover the hillsides. And when you find a fossil, you put a flag next to it, and then we can evaluate whether or not it needs to be collected. So look at all the flags in this picture. All of these flags designate a fossil or a partial fossil found on just this one hillside. And once these have been collected, you move on to the next hillside. You might notice that some of the flags are white, and you might wonder what the significance of that is. The white flags indicate that we ran out of blue flags. And so that's all that means. We just needed more flags. But once all of these were collected, we move the next little ridge over and start the whole process over again. During the heat of the day, once you reach lunchtime, we'd head back to camp and avoid the heat of the day. And that's when you clean up and start identifying all of the different fossils that you found and cleaning up the ones that really need work, especially some of those preserved in somewhat harder matrix, and then starting to think about them. This is actually me in camp, and that's actually a horse tooth that I'm looking at, a fragment of a horse lower jaw with a tooth in it. And this is where we switch over to horses. The horses in East Africa are, at least in the time period we're talking about, different from the horses in Africa or the zebras in Africa today. These are known as Hyperionine horses. It's a completely different lineage from the horses that we think of today. If you've seen the classic horse lineage that's been presented by people like Bruce McFadden, these are living horses today all over the world. And because these are the horses that are so much the focus of our own lives and that so many people are interested in, usually this is the lineage that people are interested in hearing about when you're talking about fossils. But as I really want to emphasize, there are whole separate lineages that aren't part of that line leading to modern horses. And the one we're going to be talking about today, the Hyperionine line, is this one that you see over here that originated in North America, but then spread out throughout the rest of the world, including arriving in Africa more than about 11 million years ago. In the Hadar region, you have a couple of different species, Urinator afarense, as you see here, and also Urinator hippos hasumense is another species that's known from the area. So this is Urinator hippos, which is quite a tongue twister. And you can see that these are some of the really nice fossils from Hadar that have been preserved. This is a snapshot of what the upper cheek teeth would look like. This is the occlusal surface. This is what they'd look like if you were a piece of grass, and this is the last thing you saw. And you can see, those of you familiar with horses, these are enormously complex surfaces of these teeth. There's a lot of enamel. All those ridges and wiggles you see there are enamel used for chewing abrasive fodder. Hyperionine horses do things a little differently than the lineage leading to horses today. Horses that we have today and the lineage leading up to them have taller teeth in the jaw, but somewhat less enamel complexity. Hyperionine horses have more enamel complexity, but less tall teeth, less hypsodont teeth. The specimens I just showed you were from the Hadar Classic region. The materials we find in Lady Guerraru are frequently less well preserved. This is what a typical horse tooth find would look like at Lady Guerraru, but you can still pick up the pieces and assemble it. One feature to point out on this tooth is this feature here, which is known as the protocone. And one of the distinguishing features between Hyperionine horses and what we can call true horses would be that in Hyperionine horses, the protocone is separate from all the other cusps on the rest of the teeth. Whereas if you were to look at a Pleistocene horse from North America, like this one from Las Vegas, you can see that the protocone there is actually connected to the cusps on the rest of the teeth. So that's one distinguishing characteristic of these horses. If you look at the lower teeth, you see another distinguishing feature. These are lower premolars, and then the lower molars over there on the left. And if you look at these little oval folds of enamel, these are called ectostylids. And this is a common feature in all of the Urinator hippos species known from Africa. And this is something, again, that doesn't show up in Pleistocene horses from North America or in modern horses for that matter. In addition to dental remains, we also occasionally find postcranial remains, and this gets me kind of excited. This is actually a metacarpal from Lady Guerraru, found by a grad student, Irene Smale, who fortunately took the time and effort to hunt up all of the different pieces of this find so that we have a complete specimen that we could glue together and measure. And so, Blaine, this gets back to your question. All of these specimens go back to the National Museum of Ethiopia in Addis Ababa, where they get cleaned up by their professional paleontological preparators. That's what you see happening here. And then you can compare them. You can take the new material coming in from the field and you can compare it with existing fossils. And you can also eat egg sandwiches and drink amazing Ethiopian coffee while you're working. So there, I did have a food.
We were right.
I
Eric,
feel better now.
when we look at those teeth, they are so complex looking. I'd never seen one of those on the occlusal surface like that before. Would you consider them to be more specialized then than the living groups of horses?
That's a really good question, and I'm not sure. I think it's just a different specialization, and this is something that Nick Famoso and Darren Panyak and Bob Faranek have been talking about, is distinguishing the levels of complexity. And again, with Hyperion horses, what you're looking at is just, it's kind of a way of investing the same amount of energy in the creation of a tooth. It's just doing it differently. It's kind of like front end loading the energy so that you're producing complexity, but then you don't get a tooth that's quite so tall. Whereas again, with living horses, Pleistocene horses, North American horses, you don't see nearly that level of complexity, but the teeth are taller. So they're going to wear down maybe a little faster because there's less enamel, but they'll also last comparatively longer because they're taller teeth. So it's like two slightly different ways of taking on the same task. So I don't know that it would be more specialized. I think it would just be a slightly different approach. But the teeth are beautiful to look at. It's just, I don't get over it. It's really cool. And I'm so used to Pleistocene horses that every time I look at this, I just, okay, that's really cool. I like.
And so when do they go extinct, that type of tooth?
I honestly don't know worldwide. I believe we have a Hyperion horse in North America called Nenippus, a little tiny one that held on up to around 2.2 million years. And the youngest Urinator in East Africa is from around 500,000 years, which is really interesting because the earliest record for Equus in Africa is around 2.33 million years, which means that you had almost a million years of overlap between Equus incoming into Africa and the standard established equid species, Urinator, or genus Urinator. And so if you go back to kind of the old classic literature where people talk about, oh, well, you know, Equus comes in and it's got one hoof, so it's obviously better, so it just outcompeted it. That's a long time to take to outcompete these organisms. So one of the things that Kay Reed has consistently said is she really wants to dive into that, which is why she keeps encouraging me to work this stuff up in more detail, because what happened to Urinator? Was it just competition with Equus? Was it something else? I think that still needs to be explored. But yeah, so 500,000 years in Africa.
Thanks.
All right. Getting back to that metacarpal that Irene found, this is what it looked like after it was cleaned up by Zinesh Kefalou at the museum. She's now in Germany studying. Anyway, sorry. These are plots of fossils, both taken from the literature and my own measurements of the Hadar fossils. And what has been proposed is that if you go back more than 5 million years, these horses in Urinator were relatively small. And then they increased in size through time as you reach the Hadar region. But at the very end of the Hadar sequence, at around 3.0 million years, you have a drop. And that was based on a single specimen, which is this specimen here. It's easily the smallest of the metacarpals. And so it was argued by Ray Bernor and his colleagues back in 2005 that this potentially suggested either a drop in body size or the appearance of a second smaller species in the region, based again just on one specimen. And so since our fossils from Lady Guerraru are at 2.8 million, we would anticipate that the size of our fossils would fall closer to this specimen than they would to some of the earlier Hadar specimens that have been reported from older units. And as it turns out, that's exactly correct. That's where the Lady Guerraru, that little green diamond, is the Lady Guerraru specimen. And so it clearly is plotting closer to that one specimen from the uppermost Hadar site, or Hadar formation. And so it's consistent with the overall story that's been proposed. A better fossil would be distal tibias, because if you know your horse anatomy, or I think large mammal anatomy, and generally the distal tibia is a nice solid piece of bone, and I love them because you can plant your calipers on them and get really reliable measurements. And so this is a plot of measurements of distal tibias. And so this is the entire Hadar sample. From the oldest units to the youngest units. You can see there's a lot of horses there, but they are all larger than the later material from Lady Guerraru, which is, again, demonstrably smaller and falls in the range of early Equus oldowaensis, from the Bucedema formation and also from Homo member G. And so, but just to clarify, when I talk about large and small, frequently when people hear about fossil horses, they think Eohippus or Hyracotherium, tiny little horses. So I want to also throw into this graph, these gray triangles, or excuse me, diamonds, are Grevy zebras, which are living zebras today, and they are the largest zebra known. So the Hadar Urinator sample overlaps the upper end of the range of Grevy zebras, or is even larger. So those were big horses, big animals. The Lady Guerraru Urinator and subsequent Equus were substantially smaller than Equus grevii, probably closer to plain zebras in size. And so this is, again, consistent with that story. And it also ties in with climatic changes in the region. Again, you have this area being pulled apart by tectonic activity. You have it becoming increasingly arid and opening up and forming the arid environments that we know now. And since this is Lady Guerraru at 2.8 million years, you're also right there at the onset of the ice ages when you start having these more global fluctuations in climate. And so you have that increased aridity and global change as well. So that's also proposed as one of the many reasons why humans separated out from other great apes, moving out onto the savannas as the savannas are opening up, taking advantage of those climatic changes, and eventually evolving into Homo, and then evolving into us today. So there's the Lady Guerraru horses are fitting into this narrative very, very nicely. They're dropping in body size. You see that elsewhere in Africa. They're fitting in with the climatic story, the tectonic story, the hominin story. It's all kind of holding together, which is really nice, except for these fossils, also from Lady Guerraru. That's first and second phalanx. And if you plot them against first phalanges from Hadar, you can see that they actually plot up in the upper part of the sample range. So right back at it, are we looking at two different species, a big one or a little one? Are we looking at one size variable species? We're going to need more fossils. And every paleontologist worth his or her salt is going to say the same thing, but it remains the case that we are going to need more fossils, specifically more horse fossils from Hadar, from Lady Guerraru, in order to really tease out these relationships. While that's true, I don't want to convey that it's just field fossils that we need. We also need to really go through some of the museum fossils. One of the lead geologists on the Lady Guerraru project, Chris Campesano, did his dissertation back in 2007. And even then he was saying, you know, these fossils really need to be worked up a lot more. And so we're now over a decade later, and that's still the case. These collections still need to be explored more thoroughly, and they still need to be discussed more openly. For example, this is a distal metatarsal of a horse from Hadar. And the one on top is, hopefully you can see, deeply pathological. There's a lot of extra bony growth. The articular surface has been misshapen. That's a normal metatarsal on the bottom image. So hopefully you can compare the two. I don't know what this pathology is. And I've talked to veterinarians who study lame horses, but veterinarians basically are looking at, okay, here's what happened to your horse. Here's how we fix it. I'm looking, and I'm actually throwing this out there. Anybody who wants to, who's interested, reach out to me. I'm looking for somebody who can come up with what causes this. Was this an injury? Was it an infection? What would happen that would do this sort of thing to this fossil? So if anybody out there is interested in looking at this with me, please let me know. I'd be happy to talk to you. There's also fossils like this. This is a complete metatarsal from a Hadar, and it is extraordinarily long and extraordinarily thin, which kind of caught me by surprise. I didn't expect that. I should have, because there are other earlier Urinator from Ethiopia and from Kenya that also have long, slender limbs. And I would like to plot this out as a graph and show you, except there were only 2 other complete metatarsi in the Hadar collections. And so I would have given you a graphic plot with 3 points on it that wouldn't have conveyed anything. So please just take my word that it's very long, very slender. And that makes me wonder about the adaptations that these animals had to the region as it was changing through time.
Eric, I want to make a quick comment just to remind the audience to go ahead and send in any questions you have to Facebook. And if we could go ahead and wrap up this part in about the next 5 minutes. I do have one question for you too, and that is, are you also running into fossils that are weathering out that you're then able to chase into partially articulated skeletons, or is it usually just isolated bones?
Me, no. I don't find very much.
Okay, you in the broader sense.
My search image is wrong. One example of my search image is finding a saber-toothed cat. I found that saber-toothed cat jaw I showed you earlier. I was very excited. So when I saw another similar thing, it looked like a saber-toothed cat canine, and I was practically dancing, and it was eroding out. It was going back into the hill. And one of our team members is Lars Verdelin, who is a world-renowned carnivoran expert specializing in things like saber-toothed cats. So it's like, Lars, get over here. You're going to just go ape. And he comes over, and remember, it's like 100-something degrees out there, and I'm making him hike up this hill to look at my spectacular saber-toothed cat canine. And he looks over my shoulder and he says, keep digging. And so I keep digging. It was a hippo tusk, and everywhere there. So part of that is my unfamiliarity still with so much of African fauna. And part of it is that the carnivorans are rare. That's one example. But on the other side of that, we actually did find a largely complete, I believe, wildebeest skeleton preserved. And so there was a skull nearby and then some bones sticking out. I think Irene Smale, again, is the person who found that. And we spent several days digging out an entire wildebeest. That was a lot of fun.
All right.
So yes, it does sometimes happen. I would love to have it happen some more, but the Lady Guerraru fossils are a lot more fragmentary than what you see in the classic Hadar sequence, and frequently or almost always occur just at the surface. So literally you see them like this hippo femur that you're looking at here in this picture. They're just lying there. And we didn't collect this because it's a hippo, and there are so many hippos and they're so large that we just can't collect them all. So it's probably still there. But once we are looking for are things like Lucy. And this answers your earlier question as to whether or not I got to see her. This is actually at the museum. And it also adds these fossils like the new jaw from Lady Guerraru that documents the earliest appearance of our own genus so far at 2.8 million years. But there are also still the horses, and they still need more work, and we still need more fossils. So as we look at these spectacular Hadar exposures and all those wonderful volcanic tuffs, realize that there is still so much work to do. There's more fossils to find in the field. There's more fossils to describe in the lab, and it's very exciting. To me, it's just like brand new horizons, which is why this is kind of me these days, where even though traditionally I study equus, lately I've kind of been moving on in other directions, and I'm really hoping to get more into some of the East African fossils. And with that, I'd like to thank all of these people who made it possible. I want to give a particular shout out to Kay Reed and Amy Rector and the whole team, without whom I wouldn't be able to participate in this. But there's the Ethiopian Ministry of Culture, there's the National Museum in Ethiopia, who very graciously allowed me to look at their collections. And of course, this is supported by the National Science Foundation. And with that, I think that's the end of my presentation officially. So happy to try and answer any questions you have.
All right, thank you. I know we have a number of questions rolling in, and let's go over to David and see if he can jump in with one.
Sure thing. And as a reminder, we'll remind everybody, go ahead and start asking your questions in the Facebook comments, or again, if you can't, I'm keeping an eye on Gray Fossil Site's Instagram and Twitter. Let's start with, I'll group these two questions together since they're pretty similar, Eric. Stephen asks, are there any proboscideans from Lady Guerraru? And Gino asks, have there been any hyena fossils found there?
Oh, hyenas, I should know and I don't. And I apologize. I will look that up for you and get back to you on Facebook. That's a really good question. Proboscideans, yes. And there's a story there. But yes, we find proboscidean bones. We found a beautiful proboscidean jaw, and a bunch of other stuff. And kind of like mammoths in North America, you also find fragments of enamel.
Can you tell us the story, Eric?
Yes, yeah. There is some way away from this, but I believe part of the same complex is an archaeological site where they had a mammoth lower jaw. And I mean, no disrespect to any of my colleagues in Africa, I really don't. But as I've emphasized, the fossils are sitting at the surface. You pick them up, you carry them off. With this jaw, it needed to be plaster jacketed, and nobody had ever done a plaster jacket. So there's me sitting there. They had some dental plaster, but and while it would have worked, it wasn't there. So we had to drive into the local town, Mile, and we found a place that sold plaster. So we bought the plaster. That's good, but they don't sell burlap. So we bought a couple pairs of jeans and chopped them up and used that instead of burlap for the plaster jacket. And somewhere on my computer, I have pictures of plaster jacketing an elephant tooth. If I said mammoth earlier, I apologize. When I think big proboscidean, I think mammoth, but everything we're looking at here is elephant. And so there was a very large elephant tooth that needed jacketing, and I got to show off that I actually can still make a jacket with jeans.
We can be creative, that's for sure.
Yeah.
But
Here's
yeah,
another. Oh, go ahead, Chris.
Eric, I was going to ask that question first, and Stephen jumped the gun. So that's cool. I'm really interested in the proboscideans. But one of the things I was thinking about as you're talking about this early Homo jaw, do you think, is there a hiatus between Australopithecus and Homo in other parts of Africa, or is this something that we're, as we get more fossils, we might see kind of this transition between Australopithecus and Homo elsewhere? But I mean, what do you think? Right now, probably not very many fossils to really address that, but you've thought about this more than I have.
I have, and to be honest, it wouldn't surprise me. I mean, honestly, and if I have any colleagues who are paleoanthropologists listening to this, I will probably embarrass myself and they will laugh at me derisively. But my own experience with horses is just that the more you look at them, the more they blend into one another. And so if you have just a few horse fossils that you say, oh, you've got species A, species B, species C, and then once you start getting big sample sizes, it's like, oh, there's all this overlap. And I think that is still a problem with North American horse systematics today, complicated by the addition of DNA work where everything lumps together. So I'm increasingly leery about species divisions based on small morphological differences. To me, the morphological differences of that jaw from Lady Guerraru are really convincing. I hope I captured it with that picture, but I mean, it's strikingly different from the Australopithecus material. That said, it wouldn't, the hiatus is very clearly there elsewhere in East Africa. So it wouldn't surprise me if Homo was much more far ranging than we currently think, just because this earliest jaw is present at Lady Guerraru. Part of that is because, backing up a little, I'm a latecomer to this team. Kay Reed invited me to participate back in 2014, 2015. A lot of these people like Kay and Amy and Lars and so forth have been out there since around, I think, 2004, 2005. And they were just scouring this part of East Africa, looking for different areas that had the requisite radiometric dating and the requisite abundance of fossils. And so it's no accident that they found this region and started exploring this region. It was great that Chulachu found that lower jaw, and there are other hominin remains coming out of it. I don't want to talk about them because they haven't been written up yet, but this is not the only hominin there. That said, once you get out of this region, if there are other areas that fall in the same time period that have fossils, it wouldn't surprise me if, with more intense focus there, you started finding some Homo there as well. That was a very long-winded answer, but hopefully I answered your question.
No, perfect. Thanks.
We're going to have a lightning round, David.
I don't know what that means.
Well, we've got a bunch of questions. Some of them might be pretty quick, but I'm gonna. The next one I'll go to is this question from Barbara, who says, "Sorry if I missed something, but why again are these fossils all just lying on the ground where you can pick them up?"
Because the area is so remote. If you go back to discussions of North American paleontology back in the 1800s, when they're talking about dinosaurs, it was exactly the same thing. You just had dinosaur skeletons complete sitting there at the surface. Once you start collecting them, things get a little harder. So basically, it's just because nobody had really looked in this area before.
And Eric, I think a lot of people that are watching this are from the east over here, and everything's covered in vegetation, and might not be used to the fact that, when the sediments weather out, when a sediment's weathering that has fossils in it, they will just be all over the surface.
Yeah, these sediments are just absolutely full of fossils. So if you're not disturbing them, that's just natural weathering and erosion, then yes, the fossils are going to be exposed. So that's a really good question, but that's the basic answer, is because the area is so remote.
Great. And then I have there's another taphonomy related question here from Maggie, who says, clearly there is a high quantity of fossils, but quality of preservation seems to be low. How has this impacted your data collection?
That is, again, an excellent question. I have to stop saying that, but that really is. It's impacted it because I showed you that slide of us planting flags up and down the hillside. And that's the first step. Here's what's on this hillside. The next step is looking at each and every one of those fossils and saying, is this worth collecting? Is it diagnostic? Does it have a little hook or a curve or something that will enable us to identify this back in the lab? And if it doesn't, then we don't collect it. That actually, changing the subject but not, that's the same thing we do in paleontological mitigation when you're working on a construction site. You aren't in a position to collect every single fossil that's exposed by construction. So you have to do a very quick evaluation, look at it and say, is this worth collecting? And if not, you just leave it. If it is collecting, you get it out of the ground quickly. So that background in paleontology for me really stood me in pretty good stead with the East African materials, where it's just walk up the hill and that's identifiable, that isn't, that is, that is, that isn't. And what isn't identifiable is left behind.
Very good. I've got, Blaine, were you going to say something?
Oh, I was just going to try and jump in with a couple of the questions that I see
sure.
here. And a few people asking about more of the different animals. Jim Mead's asking, of course, about the bitty lizards and snakes that might be out there.
And again, and Jim, I'm sorry to disappoint you. I should know, but let me look that up for you. I should know about that.
And Charlie Bruce.
Go ahead. Oh,
I was just going to say, and Charlie Bruce is asking about the types of carnivorans that might be coming out of there other than what you've already mentioned, and whether or not that cat is dinophilous.
My understanding from Lars Vertelen is that the cat is Homotherium. So, and apparently it's a very large Homotherium. He was quite pleased with it. And Kay was pleased with it. So I felt like I'd earned my keep that day. But
what a cool thing to find.
In the interests of time, I took out the slides, but we were working in one area and I saw this, it was white. And, you know, usually if you're in the field, you see white bone and you think modern. And so I almost kicked it out of my way, literally, and then went, wait a minute. And if you know your sabertooths, you know that they've got that flange on the front end of the jaw. So you have the lower end of the jaw and then it dives down to make that flange. And so suddenly I realized there's a carnassial, there's the jaw, there's the flange. Oh my gosh, I've got a saber-toothed cat. I think I said something a little stronger at the actual time. But I was extraordinarily pleased with myself for that, and then collected it, took it back to the camp, and, you know, started washing it to clean it up, and it completely fell apart. It was just sediment holding it together, so it fell apart in chunks. And so that picture I showed you was after spending an entire afternoon gluing it back together. So there was, in addition to being a nice find, it was also a bit of a labor of love. And it now resides at the museum in Ethiopia.
I've got a couple of questions here about stable isotope work. Greg McDonald asks, has stable isotope work been done on the Hyperines? And Grant Boardman asks more specifically, are there isotopic differences between Urinator hippus and modern equus that might explain the extinction?
I don't know that that work has been done that thoroughly. I don't believe that it's been done with the Lady Guerraru sample, but there are other sites in East Africa where there has been stable isotopic work done, as well as mesoware work done. But what you're seeing is pretty much what you would expect to see, is that at least the late Urinator hippus is a grazing animal. In fact, I think Urinator hippus, it's endemic to Africa. All of the Hyperion horses in Africa are Urinator hippus. But in 2019, it was reported from the Indian subcontinent by Advait Jukar and his colleagues. And I believe that they did some studies, some isotopic studies in there that documented that, in fact, it was a grazer the same as equus. So yes, the work has been done. Yes, the work needs to be done on the African material, but now you're in the same position where all of the, and this is not a criticism, it's just stating the state of affairs, all of the fossils go to the museum in East Africa and they stay there. So I couldn't borrow them, bring them here, isotopically sample them and send them back. I would have to bring the sampling equipment to Ethiopia, and they would have to sign off on that in advance, because it is consumptive sampling. And so if we were to do that, we would need to write up a grant, we need to get approval, we would need to go back to the museum and collect those samples. And I think it's something that needs to be done that's really worth doing. And I would like to tie in some mesoware analysis with that, but that remains to be done.
All right, as we are in the last few minutes of our program here, here's one more great question, and then I actually have a follow-up to this question. I'm glad to see this question get asked. Mason asks, what's your advice for young people going into the field?
Oh, I guess my honest answer, and it's not a good one, is passion. I loved this stuff as a kid, and yeah, I got away from it when I went into high school, but I was still volunteering and studying fossils not studying, but preparing fossils at the tar pits on the weekends. And I loved it. And that focus, if you have it, will probably carry you through, because you would need to, honestly, I don't know how old he is. If you are a grade school or high school person, you might not have access to anatomy classes or things like that, or osteology classes. But I would argue that one of the things you could really study that would help you is reading and writing. And I know that sounds very basic, but those are the biggest tools a scientist has. And then just look at what your options are in college when you get to college. But for me, it was just interest and love and passion that carried me through. And I see that with so many of my fellow paleontologists who have been doing this since they were little kids. And you show them a fossil and they get all excited about it, and you show them a new fossil and they start dancing a little bit. And I don't think that happens in a lot of other lines of work. So if you have that interest, that's a great thing to keep in mind. If you have a local museum or a local university that has a museum in it, reach out to them and see if they have volunteers. So much of what I do, I even mentioned in terms of identifying a saber-toothed cat in the middle of East Africa. That's stuff that I learned when I was a volunteer at the tar pits. And so that can really set you up. Other than that, going to school and working on a degree would be great. It would depend very much on what you want to do. And maybe I'm getting too into this question, but there are some people who want to do research. There are some people who want to work in the field. There are some people who want to just put broken fossils back together. What you want to do is going to determine what kind of schooling you want, how far you want to go towards various kinds of degrees. If you want to do research, you're going to need a PhD. If you want to do preparator work, you might not, but that's where getting experience with fossils at a local museum or university would be really important.
Great. Thanks so much for that answer, Eric. It looks like we've got a few young people having a conversation in our comments about getting into paleontology. And then I had a follow-up question.
Okay.
One of the things that really strikes me about this work that you're talking about, and this is something we haven't seen all that much of on Paleo Talks, is that it's international, is that you don't just get the excitement of digging up fossils, you get the excitement of flying halfway around the world to dig up exciting fossils. So here's a question that our young people and maybe our not so young people might be interested in. What would your advice be for people who are looking to establish international connections and travel across the world to other countries to collect more fossils and do their work?
Wow, that's a really good question. Paleontology is the reason I travel. I mean, before the international stuff, just nationally, I grew up in Burbank, and we didn't go anywhere. You know, a huge trip would be to Newport Beach down in Orange County. And suddenly when I started doing paleontology and got involved in the Society of Vertebrate Paleontology, which has annual meetings every year, and none of them were being held in Burbank. They were all being held, you know, in other places, frequently in other states. So suddenly I get to travel and go to all these other states and places that I'd never visited. And then with this opportunity, yeah, because you're absolutely right. It's not just Ethiopia. It's all the stops the plane makes along the way. We stop in Germany and places like that, or once Ireland. So you do get out. I would look, if you're looking for that, look at universities that already have that international component, that are already working in other areas. And there are a number of them. Again, Arizona State University, it's in Arizona, and yet they're working in East Africa. And you can certainly find, depending on where you are, any number of other institutions that also work internationally, and then reach out to the local institutions that have those connections, and also look at what they are studying, and see if that might give you a door in, because it's wonderful. I mean, I do like Burbank, and now I live in Bloomington, and I like this, but it is an amazing world out there. And I think travel is one of the huge perks of being a vertebrate paleontologist. So whether it's nationally or internationally, take a look around and see what's open.
Fantastic. Thanks so much, Eric.
Yeah, thank you again, Eric. This has been fantastic. Really enjoyed it. And if you wouldn't mind sticking around with us afterwards, we'll talk a little bit more after the recording stops. And also, if you can look at the Facebook and maybe answer any of the questions that we didn't get to. that'd
Okay.
be appreciated as well.
I will do my best. And let me just say thank you guys for putting these presentations up week after week. I know I look forward to them, and I think any number of other people do too, particularly over the past year with the pandemic and everything going on. This has been a refreshing and helpful and informative way to end the week every week. So thank you so much for what you're doing.
You're so welcome. And again, thanks for joining us. It's been a great presentation, great way to start off 2021. Thanks again.
All right.